Use the mic?
Yep,
Okay.
sorry, make everybody use the mic.
Is that better? Yeah. Okay. Morning, my name is Charles Mistry. I graduated in 1995 from Jim's lab, and I currently teach at Butte College in California, and I'm going to be chairing the session on community ecology, which is our first session today. Before I start, though, I do have a role in addition to moderating the session. I'm here to remind you that in spite of the fact that you might hear the words rodents and scaling and wheel many, many times today, a couple of Jim's earliest papers were actually on bats. And that's important to me because I study bats. And so he might have deviated off that path quite a bit, quite magnificently, but he did have this early interest in bats. So today we're going to start off with a few sessions. There we go. Okay. And our first talk is by Dinah Davidson, and she's going to be talking about tropical arboreal ants. And to paraphrase a popular song these days, she's got passion in her ants and she ain't afraid to show it.
Put this around here. Okay. Can you help me move? Oops. Okay, at a gathering of macroecologists, it takes a lot of fortitude to put up the world's natural history, but I thought I would do it myself so that it couldn't be aimed at me afterwards. I think that over the course of my career, I've realized that take a big dose of natural history is helpful now and then. So, as many of you might know, I worked with Jim on the Portal project, and before that, Marana. But soon afterwards, I realized that I had to shake the association somewhat to grow on my own and to establish a reputation on my own. So Jim hated the tropics because of the humidity and the seed ticks and the botflies and the lice flies, etc. So I ran down to the Amazon and spent a number of years there, and then went even closer to the equator over here at Kuala Bela along Brunei. And there we had leeches and poison ivy trees. So that was safe to be that distant from the desert. And, I want to start out with just a few generalizations about tropical arboreal ants that I hope to debunk to some extent here. The willingness of tropical arboreal ants to feed at baits is a good indicator of their natural diets and resource bases. Well, there's a whole volume on cephalotes that debunks that. A lot of ants will take sugar water and tuna and things like that, but these are specialists on pollen. Most common species are dietary generalists, for which abundance and diversity of both resources and consumers are adequately modeled in relation to productivity and temperature gradients. Resources don't vary idiosyncratically with such gradients. I think the resources I talk about today would. Generalized and similar diets across ant species enable modeling of most species as potential competitors of one another. That is, there's not a lot of guild structure. Ants with specialized diets, cephalotines which eat pollen, and cephalotines which eat pollen, pseudomyrmecines which eat fungi are rare and can be ignored. Common tropical arboreal species feed mainly as predators and scavengers, fueled by plant extrafloral nectar and insect exudants, honeydew. The effects of tropical arboreal ants on plants are therefore mediated mainly through deterrence of or predation on herbivores. Well, as a first look at this, we use stable isotopes as a technique. And as many of you know, nitrogen comes in two forms in the natural world. N14 is slightly heavier, N15, which is rare in most cases, but it increases proportionately with trophic level as lighter N14 is driven off as waste. Predators are therefore N15 enriched relative to herbivores. That's a good generalization. You are what you eat plus 3 per mil was something that was kicked around for a long time, but that's not always true. So these represent data from a paper in Science on Peru on the left and Borneo on the right. What we did was to gather up a lot of ants, get their nitrogen isotope ratios, and calibrate. Here are plants, trophobionts, which are ant-tended hemiptera, chewing herbivores and predators. And what we found was in both forests, there were a lot of things that fed sort of as herbivores, even as plants in some cases. So they had low, unusually low nitrogen 15 values. And those included pseudomyrmecines, which are pollen and fungus feeders, myrmecines, cephalotines, which are pollen feeders, dolichoderins, which recycle nitrogen in their guts and then have autocoprophagy and anal trophallaxis, so the light nitrogen is getting recycled differentially, but also formicines in both the New and Old World, and especially campanotines, which feed as leaf grazers, so they would be here and a lot of them over here. So they could be eating wound exudates, inconspicuous extrafloral nectars, etc. The group I'm going to talk about today is going to be probably its own genus at sometime soon, Colobopsis, and now in Campanotus. And there are a lot of species at where I worked, and across Asia and Melanesia. Probably 11 of ours at least are unnamed, which makes publication a little difficult. All forage strictly diurnally by grazing adaxial top surfaces of leaves, old epiphyll covered leaves, not the young extrafloral nectar leaves that where most ants feed. They take extrafloral nectar and sugar water, but they don't tend to stop feeding hemiptera. They have a larval structure which mimics that of pseudomyrmecines, which is a pocket here. It's convergently evolved, and a long, thin neck, and the larvae feed out of this pocket where the workers deposit food. These ants are very common, so they're probably not pollen feeders. They might be fungus feeders. In a subset of these species, exploding ants, workers deploy autothesis, or intentional self-sacrifice in defense of foraging territories far from the nest. And this sets them apart from other social insects that have autothesis. They usually are defending the nest and the queen and the brood. Across the phylogeny, if we get that far, their derived nesting habitats are more pathogen-plagued, so increasingly pathogen-plagued nesting sites. Their codominance with species in this Polyrhacus, no specificity in these relationships, but they always forage together and they codominate and defend territories together. It's hard to map territories in 3 dimensions in the tropical rainforest, but all these colorful dots are the species of Colobopsis. These are territorial Myrmecaria, these are territorial weaver ants, and so there's pretty much a continuous territory spacing, and the spacing is relatively short between them, 20 to 40 meters, and most of these species will forage over 100 meters. So we had 16 territorial species, and tropical ant ecologists once thought that cavity nesting ants would have to be rare because there's not enough available suitable cavity space, but these ants are very common, but they're also cavity nesters. Okay, so the interesting thing about them, their morphology, is that they have the mandibular gland reservoirs, normally confined to the head, all the way through the body. You can see it on a callow worker here. They have inside some beautifully colored goos, which are pH-specific colors. They've got phenolic rings, phenolic acetogenins, and they're essentially litmus paper with legs. Here you can see it through the exoskeleton. And hypertrophy of those glands costs them something in what they're able to take in liquid foods. So all ants groom, and they groom particles into an infrabuccal cavity, and then they are thought to spit it out. I think we're going to find more and more that they don't spit it out. These, in fact, digest them in the head. Here's one that's still black, sort of black mashed potatoes, and down here it's partly digested, and here it's turned totally into fat. Large holes in the head indicate that they were once there. There is a membrane around them as they're being digested, and the mandibular gland products, this one from our species barbecue with red goo, is shown there. They drum on leaves and then they feed, so that we think they're drumming on leaves. The anterior openings of the mandibular glands apply to the leaf goo, and then they eat what they're eating with the goo. And my colleague Tappy Jones believes that these phenolic acetogens behave as food preservatives and maybe protect the ant also from infection because they're weak antibiotic activity. If you look through the phylogeny, which we may see in a minute, from the lower part of it to the middle to the highest part of it, they're taking fungal spores, mainly fungal spores, but you also see microarthropods in the head, and the mandibles seem to be of the species that we opened to get this, so they're consuming their own worker bodies. Pollen grains, and there's also and the mandibular gland product, seen here again, also contains protein, except where it's caustic chemical. And we think that might have to do with digestion. They're mixing it up in their digestive enzymes there, and there are reasons, the way the ant gut is built, why they do it this way and not some other way. There's also an imposter species which forages with 4 of our ants with M. cresol as the compound in their mandibular gland. And they have the same diet. And there it is there. It imitates the behavior. It pounds on the leaves. It has no chemistry, volatile chemistry, so it can sneak around on the leaves and never be detected. And it's pretending it's treating the leaves. The fact that this specialized parasite treats the leaves like that suggests that there may be some farming going on by the Colobopsis, and it's parasitizing that resource. Anyway, when pellets are processed in the oral cavity, they produce lipid-rich material, and in the absence of larvae, it buds off and it goes down the esophagus. This is anterior, this is posterior. When I took them back into UBD campus and they didn't have sugar available, they couldn't process them as well, so the partly digested stuff they stuck in the larval pocket, and it shows up, I can't see it too well from here, but there's spores in that. So they are feeding it to larvae. When we looked at the DN15 value of these pellets, and believe me, that's a lot of pellets, we found them to be the same as herbivores. So fungus eating on the phytoplankton, they're like herbivores. That's a little too high to account for the fact that Colobopsis has the same ratio, but we think, again, there are 2 kinds of nitrogen recycling, preferentially light nitrogen being recycled. Yeasts are also involved in this system, and we think they're really important. There are a variety of them, and they're in the worker crops, and they may be processing spores as food here and in other Camponotus. They've been identified in some Camponotus from Oregon. They might be doing polysaccharide reduction from them by the ant. Ant digestion may help to form that envelope that encircles the pellet. They can have antibiotic activity to reduce the danger of infection for the workers. They might be secreting enzymes, though they don't secrete exokinases. They could contribute to some form of pellet digestion, detoxification of fungal spores, or to digestion of hemicelluloses ingested when leaves are scraped. And we have 2 species where we suspect they're actually eating plant material and processing that as food. Variation in yeast species and mandibular gland product pH may be correlated with one another and with interspecific differences among ants and mechanisms of food processing. So we think that leaf grazing ants may affect plant fitness. Some colleagues of mine in Austria are identifying organisms from the pellets molecularly. They find Fusarium, which is a plant pathogen, often plant pathogen, to be common, and also Trichoderma species to be common on the leaves, and those can be protective of the leaves. They're mycoparasitic. Quick aside, other ants in this same rainforest eat fungi. This is a case where it's the only case outside of the Atani, the leafcutter ants, and the only old world case where they're growing fungi for food. This is a Colobopsis nest. The ants go in and get water, and they pee it all over the substrate, which is riddled with Fusarium, and then they eat the product, And it's patchy, the things that they eat are patchy, and we think that comes from, there's a lot of insect cuticle in there, and when that degrades, they pee on it, and there are hydrophobic proteins in the structures that support the macroconidial spores, they're aerial hyphae, and they may be, all that water added may cause production of more spores. Another aside, these are ants that don't leave the leaf shadow of their host trees, and we suspect that they might be involved in some kind of interaction with the plant fungi in that case too. And this one, they have compounds that induce plant defenses and change those. They're changing here. They just use their mandibular glands over the entrance of their nest, and it grows out like this. So they may have some role in imitating plant hormones and plant signal chemistry. Okay, autothesis. The ants are, as I said, suicidal. Some of them are. That's a derived territorial fighting mode. Minor media workers only commit suicide. The soldiers don't. Lower in the phylogeny, a basal trait is that they fight with soldiers, major workers, and the heads in this group of Colobopsis are longer than they are in other Colobopsis, maybe to keep the goo from one another in territorial fights. It's triggered by, behavior is triggered by leg grabbing, and the greatest leg grabbers in the forest are Ecophylla, which are predators as well as territorial against these species. You suspect a low-density resource when they're territorial to the extent that they are, and extreme territoriality may affect unusually high investment, like farming in territories, but also especially high threat by invaders, contamination with foreign yeast or other microbes. You find aggregations in the field sometimes. These were from a palm leaf, and there were hundreds of coupled pairs of the same species. They were in an uncharacteristic host tree. One colony was. It was too hard for them to work on it, and from the inside, and they went downhill to try to find a better nest, and they had a big fight on their hands. What they do is they try to wrap themselves. When they blow up, they exude mandibular gland compound out of the anterior openings, and they blow up the gaster and attach themselves to the top. They try to wrap themselves around the ventral side of enemy. This works against much bigger enemies. And the dorsal wrap sometimes works. Sometimes they lose 2 workers in a fight. Anyway, it's a very effective means. 84% of the time, suicides were successful in getting rid of them. So here's one small ant, yellow goo, finds a major worker of barbecue, quickly dispatches it. And you see, it detected the footprints of barbecue on the leaf, and so that's why it was searching for it rather quickly, and they end up in the leaf litter, and they're no longer a threat. The body size spectrum in this rainforest is somewhat bimodal, but if you add these species in, it's highly unimodal. It affects the variance marginally in the size distribution of arboreal ants in the forest. And so you can see that these ants occupy a fairly narrow range of body sizes that could have something to do with phylogenetic inertia. We also think that it only works in those sizes, and there might be ants that are in the middle of the spectrum may need special defenses to counteract having smaller ants and larger ants as potential competitors, though these really aren't. So it might just have to do with fighting strategy. The smaller they get, they back away from this kind of suicidal fighting, and they can't do it effectively. Food and nesting space, are both limiting sometimes for ants, and they're essential, non-substitutable resources, so they need nests as well as food. Factors affecting availability of nest sites, nest longevity, and protection from pathogens, especially for ants that bring back fungal spores to their nest. This is a phylogeny from Steve Cook's thesis, not yet published otherwise, and you can see the colorful lines are nesting habitats. So these are nest only in the canopy. These nest also in the subcanopy. We're not sure about canopy nests because can't see them always. And these are the two smaller species that have gone back to the canopy. And these species have M. creosole, and they occupy, brown means that they have, they nest in dead wood as well as live wood on the forest floor, so that's very pathogen-plagued. The chemistry evolved prior to the hypertrophy of the glands, which makes sense, and then as the amount of compound increased, they had autothesis as a fighting mechanism. Let's see, I have to go through this quickly. pH indicating product colors are over here and don't show too much of a pattern. Although there's some other patterns I can't talk about. We built 60 artificial nests, put them out. We could harvest the ants and look at the fungi on the nest wall, and then put them back. We compared, using molecular probes, we could compare the microbes that were in the ant nests, ant Colobopsis occupied, other ants, and empty nests. So the results were in occupied nests, any ant nest, diisotrophic nitrogen recycling Burkholderia bacteria were overrepresented relative to unoccupied nests. Mycoparasitic or cellulose digesting trichoderma fungi were overrepresented in the nest wall of taxa with M. creosole, so that they tolerate M. creosole. Burkholderia actually breaks it down. Species investing most in M. creosole have extremely hard nests. They live with both Burkholderia and Trichoderma ricei, which is the one they use for commercial cellulose digestion. So the ants are using that too. The enhancement of the nitrogen environment should speed microbial cellulose digestion, leaving highly recalcitrant lignocellulose, which few microbes can degrade, and the nests are so hard you can use any tool and implement you can find and you can't break them open. I'll skip that. So what have Colobopsis and others taught us? Okay. As in other cases, ants that collect nectar can be specialists on something else. We need a better definition of feeding guilds to understand competition among tropical ants. Common ants are not always dietary generalists, and their abundance and diversity may reflect idiosyncratic variation in productivity. These ants drop out of the community pretty rapidly when dry habitats like recovering forests, and they're very slow to recover. And we had one major drought, and they all stopped foraging, and their colors of their goo changed and so on. Ecologically and behaviorally dominant arboreal ants don't always feed as predators and scavengers. And the effects of tropical ants on plants may be mediated through plant microbe interactions. And there should be a gradient of that, with this being much more important in equatorial forests. And we see that there are more grazing ants. New questions arise. This is a very difficult thing to study, so even where fungi have been recognized before as important resources of ants, rarer ant species, it's poorly studied because you can't get up in the canopy and sample microbes in ants. So we don't know the specificity. We don't know whether the microbes harm or benefit the host in general. That varies with ant species. Are there implications of the stem nesting thing, the stem microbial flora for myrmecophytic plants? How rapidly can epiphyll grazers recover in secondary forests? Very slowly. And then there may be, other ants eat microbes, but they don't have the processing equipment that these ants do. And what we've seen is that they recover very slowly from our collection. And so they might be the oil birds of the ant world. I'll explain it later if anybody wants to. So my evolving look at tropical rainforests. First you go to rainforests, it's so overpowering, the diversity and so on, that you look at individual species. Then you look at interacting co-evolved systems. You branch out from that, and you know ants well enough to do stoichiometry. And everything in the forest looks to you, you're going around going carbon, nitrogen, carbon, nitrogen, carbon, nitrogen. But now I'm going around going, I see a pattern here where these ant colonies come together and all the branches are falling off the trees. Their yeast are fighting. Who knows? Too many men and no women at all at this research station. My collaborators. And I thank Jim and Astrid for turning me on to natural history and the excitement of science. Sorry to take so long.
Thank you. I know we're running behind, but if someone has a really short question for Dinah.